Good evening. Thank you for joining this third session. So today we're going to cover the frontal lobe, the motor and sensory system, and we're going to give particular emphasis on the parcellation map of these areas. But before we continue, we're just going to review a case to see how practical connectomics can be applied. So this is a case of a 40-year-old executive in a prominent bank, initially running multiple teams. And then in January was diagnosed with COVID, never hospitalized, was feeling pretty ill for 4 to 6 weeks. But what they noticed when the illness finished is that they were severely impaired in terms of their executive function. Working memory was a problem and concentration was a problem. As you briefly saw, the structural MRI was completely normal. So at that point, the patient came to us and we decided to analyze their connectome. And what we could see instantly, although this patient had a normal structural MRI brain, there were multiple connectomic abnormalities in their central executive network in particular, as well as their limbic and default mode network. And these abnormalities you can see in red and blue. Earlier in the week, I had the pleasure to discuss the case with Mike. So it's a recording from earlier in the week, and he'll take us through his approach to this connectomic.
First step, so we want to make working memory and concentration better. First step, of course, is look and see is there any obvious old school brain damage, because it's like these scans don't provide any information, but nothing obvious. So at least at a glance, if the hippocampus are still there. Next question we would look at is central executive. Say, is there some obvious problem? And I mean, nothing that I would write home about. Again, there quantitatively could be problems with these networks, but looking at this real quick, looks fine, basically. So going to anomaly detection, let's just look at the CN. That's a good thing. I mean, I've seen worse, but obviously there's two potential targets that are symmetrical. T1M, that's often a target we find. Let's kind of look at this compared to the limbic system, not that interesting. Subcortical, not that you could build another case for TE1M, obviously on both sides. Let's try just something more complex, like multiple demand. I don't see a whole lot there, to be honest. So, she's got things you could target. But, again, these aren't necessarily the biggest, most obvious things. T1M. Let's just, let's put the salience network into the mix. Nothing exciting. Medial to temporal. Yeah, so, I would, at first glance, I would say TE1M bilaterally would be kind of my best target for her. She doesn't have the usual, ADV, has a couple things, but she's not really an anxiety patient. You can come up with other things, but that kind of shows up across networks and things like that. That's what I think.
And Mike, would you see TBS both of them?
Yeah, that's probably my best bet on this. Because again, it's hyperactive. And I don't see any argument for being hypoactive on it. Again, you can look in other places, but that's how I would do it.
Okay. It was just interesting to us as a COVID case, because we're getting a lot of patients that are presenting with similar problems, and yet their structural brain scans are normal.
Yeah, of course. Yeah, unless they had a stroke, we know there's probably some subtle. It's like a vasculitis, right? Ultimately, not that it's vasculitis, but something along those lines. So you could see diffuse injury and things like this. Again, it's one of those things we don't know. We don't know.
Thank you, Mike.
Okay, so we'll continue on to the frontal lobe. The frontal lobe roughly occupies about 1/3 of each human hemisphere. And when you look at its essence, it's got short and long connections. And the reason for this is that the frontal lobe wants to access sensory information, wants to access memories, and our internal state. And the whole idea is to integrate all these different aspects and then exert control over regions that plan and execute motor function. Of course, it's also responsible for cognition and behavior. When we look at the ultrastructure of the cortex, essentially it's a 6-layered cortex. And what's interesting is anteriorly, the granular layers, so 2 and 4 are more prominent, but less prominent so when we're looking at pyramidal cells. Whereas the posterior parts have less prominent granular layers, but more prominent pyramidal cells. And that makes sense if you think of the corticospinal tracts with their big bed cells projecting motor output. When we look at it from an evolutionary point of view, and we'll touch on this a bit later, the prefrontal cortex is one of the 2 regions which has significantly changed when we compare our brains to lower non-human primates. The other part of the brain being the inferior parietal lobule, which doesn't exist in lower non-human primates. So to describe the frontal lobe, I'm going to go back to classical anatomy because a lot of what we're going to talk today is pinning the connectomic information that we've learned over the past few weeks onto an anatomical map that we understand. So the ultrastructure of the brain is on the lateral surface, a superior frontal gyrus, a superior frontal sulcus, a medial frontal gyrus, inferior frontal sulcus, and an inferior frontal gyrus. On the medial surface, we have the superior frontal gyrus, the cingulate gyrus, separated by the callosal sulcus and the cingulate sulcus. And inferiorly, we have the gyrus rectus, separated by the olfactory sulcus, and then the orbital gyri divided by a H-shaped sulcus. What's interesting is when you actually look at the actual configuration of these sulci and gyri, and older studies spanning a few decades back looked at identical twins, and they wanted to see if you're an identical twin, does that mean that the surface structure of your brain, the gyrus and the sulci, are they identical? And the answer is no. So these 2 brains are from identical twins, and you can clearly see that there's huge variation. So if we follow this middle frontal gyrus on the bottom brain, you can see that it's clearly different from the brain on top. And that goes to show that the brain is shaped not only by genetics, but many epigenetic factors. And this creates a problem with nomenclature because everyone's brain is different. So when you're looking to study brains in details, how do you actually know that you're comparing like for like? And the answer, of course, is going back to the Human Connectome project. We talked about the Glasser Atlas, which used machine learning, looking at prior atlases, cortical thickness, myelin, and functional MRI studies to break down the brain into 180 areas. And ultimately, what we're going to do today is look at the frontal lobe and the motor and sensory areas and try and make sense of some of these areas. It's exhaustive, it's encyclopedic, so don't feel depressed if a lot of these areas don't make sense. Ultimately, it's just almost like a safari, just going through the different areas so we can get some broad idea of what the areas involve, but you don't need a detailed knowledge to analyze a patient's connectome or make a potential treatment plan. Ultimately, the biggest factor to consider here is understanding the brain networks and what they do, because when we're analyzing the connectomics, we're looking at all the different networks. But what's very interesting is when you go into these postulations in detail, the authors of the Glasser publication actually said that to describe the dorsolateral prefrontal cortex as a term is so overly simplistic that the nomenclature almost becomes meaningless. And this has its basis in research when you're really trying to get into details of understanding what cortical parts do. Okay. So we're going to break down the frontal lobe into a lateral frontal lobe. And we're going to start in this macroregion. And the lateral frontal lobe is composed of a frontal pole, the superior frontal gyrus, the middle frontal gyrus, and the superior frontal sulcus, and the inferior frontal sulcus and the inferior frontal gyrus. The first region we're going to look in this macroregion is the frontal polar region, which has 6 parcellations. We've got 4 parcellations that are composed of the 10s. So 10 pp is the most anterior parcellation, then 10 dorsal, posterior 10p, anterior 10p, and then we've got 2 47 parcellations, anterior 47R and posterior 47R. The connections for these parcellations is the forceps minor, and that makes sense. If they're situated in the frontal pole, the forceps minor is going to connect them bilaterally. But interestingly, they also connect to the visual cortex through the IFOF, the inferior frontooccipital fasciculus that we discussed last week. What do? Ultimately, the 10 areas are activated when there's increased complexity of working memory tasks. So the more complicated something is that we're working on, the more active these areas are. The A47R and the P47R increase when the complexity of the words that we're using. So the longer the words that we're actually using in a sentence, the more active this area of the brain is. The next region we're going to go through is the superior frontal gyrus, and that has 4 parcellations, 9A, 9P, 8AD and 8BL. And you'll see there's a lot of 8 parcellations on the middle frontal gyrus and medially as well when we look at other areas. Connecting this area again is the forceps minor and that genu area of the corpus callosum, the IFOF connecting the frontal lobe to the occipital lobe, and the frontal ascending tract or FAT, which connects the superior frontal gyrus to the inferior frontal gyrus. When we're looking at function, the 9 areas, they're responsible for maintaining a behaviorally relevant working memory. The 8 areas, they're very important in spatial working memory. And what 8AD and 8BL do is they integrate peripheral vision and audition in order to create a spatial map and integrate that with our spatial working memory. We'll see 46 on the middle frontal gyrus next, and we'll also learn that 8 and 46, when they work together, what they're doing is they re-establish executive control during automatic behavior. So if someone was juggling a soccer ball and just making that automatic, and then someone shouted behind them and they had to turn the attention, those areas would be trying to perform that task. The next region we're going to talk about is the medial frontal gyrus with the superior frontal sulcus, and that has 8 parcellations. We have 8AV and 8C. We have the 946 areas. We have 46 and S68 and I68. These ones are in the superior frontal sulcus. Interesting, when we're talking about TMS, these parcellations come up a lot. So 46 is thought to be the classical TMS target, according to Fox's studies from Harvard. And 8AV, we've noticed increasingly lately, is hyperactive in patients with anxiety. Connections-wise, again, we connected via the corpus callosum and the genu, part of the FAT. And now we're starting to integrate the superior longitudinal fasciculus, that huge tract that connects the entire brain around the sylvian fissure that we talked about last week. So if we look at function, the 46 and the fused 946 areas are responsible for goal-directed higher order cognitive processes. 8AV, like all 8 areas, is involved in spatial working memory. And what it does is interprets complex visual information and guides our attention toward that information. 8 and 46, we talked about reestablishing executive control after automatic behavior. And again, these 2 parcellations in the superior frontal sulcus are responsible for maintaining spatial information. The next area we'll talk about is the inferior frontal gyrus. And parcellations in this area are 8. And we've got the 44 Broca's area, 45, 47L, and 47S, essentially all involved in language processes. And then we've got the inferior frontal sulcus parcellations, IFJP, JA, SP, SA. And interestingly, these parcellations are actually in a sulcus, they're not in a gyrus. And that's what connectomics has taught us, is that many of these parcellations don't follow a gyrus. Just because we can see it doesn't mean that's how the brain is structured. So lots of them traverse sulci and gyri. Connections of this area is the superior longitudinal fasciculus with that arcuate fasciculus complex, and makes sense because the arcuate fasciculus connects Broca's and Wernicke's areas. Again, the IFOF connecting the frontal lobe to the occipital lobe is connects these parts of the brain. And the unsnate fasciculus, that tract that connects the inferior frontal lobe to the temporal pole, also connects these parcellations. Now, what do? So if we look at the inferior sulcus parcellations, they're very important for retrieving auditory memories by connecting to parts of the brain posteriorly. They're also very important in putting together a set of instructions when we actually hear someone verbally telling us what to do. So they break down that information and make some instructions for us to do. And the IFJ parcellations are thought to be important in parallel processing, in sort of creating a crossroad between bottom-up and top-down processing of the entire lateral prefrontal cortex. As stated before, and lots of us will know that the inferior frontal gyrus is responsible for language and predominantly through Broca's area. Interestingly, 47 has semantic roles, so not just motor speech. So fMRI studies have found that it's important for some of the semantic aspects of talking. And on the right side, so on the non-dominant hemisphere, it's also responsible for cognitive inhibition of working memory. Okay, so the next part of the brain we're going to explore, which is extremely complex, and if you remember much about the default mode network we talked about, which connect the anterior cingulate complex to the posterior cingulate complex and the interparietal sulci, and that was responsible for internal thinking, assessing our internal state and imagination. It's obvious that the parcellations that link up to this network are going to be fairly complex. To break it down, we're going to look at the cingulate parcellations. So 33 prime is part of the inner row. There's a middle row, and then there's an outer row. Then there's a medial superior frontal gyrus, and then there's the orbitofrontal complex. Okay, so if we look at the anterior cingulate complex and we look at the inner row, there's only one parcellation, and that's right above the corpus callosum, and that's 33 prime. As expected, because the cingulate bundle travels along the cingulate gyrus, it's connected to the cingulate bundle, which goes all around that Papez circuit. And its function is to coordinate our autonomic, endocrine, and visceromotor responses as guided by our emotions. So if someone's getting blood pressure, hypertension because they're stressed, or if someone's getting butterflies in their tummy because they're in love, it's probably got to do partly with this parcellation. Now, if we look at the middle row, we've got 6 parcellations, 24 DD and DV, important in motor control. Then we've got a posterior 24 prime, an anterior 24 prime, and then a P24 and A24. If we look at their connections, again, as expected, they're going along that cingulate bundle because all of this is part of that cingulate lobe. And they're also connecting bilaterally to each other through the body of the corpus callosum. Now, if we look at the function, it does get complex, but essentially 24DD and DV involved in complex motor planning, 24DD for the lower trunk and DV for the upper limbs. So lower limbs, DD, and DV for the upper limbs and trunk. P24 prime is responsible for a movement response selection during very complex tasks. And A24 prime is responsible for word or sentence selection during cognitive tasks. So if someone is quick-witted and they've got quick responses when people are addressing them with problems, it's maybe this parcellation that's giving them that ability. Finally, we've got P24 and A24. What P24 does is it coordinates conscious eye movements with complicated finger movement sequences. So I like to think of this as the parcellation that potentially I use when I'm doing complex brain surgery, dissecting an artery off a tumor or an aneurysm. It's sort of integrating complex hand movements with my ability to see what I'm doing. A24 fits more with the default mode network. So it's an affective division of the anterior cingulate cortex. And it's responsible for assessing internal and external states and linking that to our emotional expression. Now, of the three anterior cingulate complex rows, we're looking at the outer row, which also has 6 parcellations. So we've got P32 prime, anterior 32 prime, D32, P32, S32, and then this 25 in the subcolossal area. Connectivity-wise, no surprise again, we're connected by the cingulate bundle going into that Papez circuit and the corpus callosum linking up both sides. Now, if we look at the function of these parcellations, again, fairly complex. So P32 prime is involved with tasks requiring attention for linguistic and sensory information. A32 prime, what that does, it's always evaluating what's important to us. So it's guiding our behavior by looking at our internal motivation, our anticipated outcomes of a certain situation. It assigns reward values and encode errors. And what this does is it allows us to focus our attention towards what's important to us. What's important to us is likely encoded through this parcellation. So it's very complex in terms of what it does. D32, that assigns value to social information. So it's probably the parcellation which decides if it's a good idea to get up during a wedding speech and have a talk or not. Then P32, again, it's sort of integrates information on a social scale. S32 sitting here, it's connected widely across the entire limbic system. So it has very wide connections, and it's probably a higher order hub of the limbic system. Again, integrating all of these functions that we talked about earlier for some uniform output. Finally, 25 in the subcolossal area is very interesting. It's a parcellation that's been shown to be involved in maternal-infant interactions. So there's very early interactions that neonates have with their mothers elicit a response in this parcellation. Okay, moving on the medial surface, 2 more regions. So we're looking at the medial superior frontal gyrus. So that has 5 parcellations, the SCEF or SCEF, 8BM, part of the 8 complex, 9M, part of the 9 complex from the superior frontal gyrus, and then the 10R and 10V, which is a continuation of the 4 10 parcellations on the anterior pole of the frontal lobe. Connectivity, again, we connected to the corpus callosum, the forceps minor, and the body as we very close to the midline. There's connections via the FAT to the inferior frontal gyrus. And then we've also got connections of the frontal pole with the temporal pole through the uncinate fasciculus. What do? SCEF is actually a higher order oculomotor center. So it almost makes an oculomotor map of our entire environment and looks at all potential options. And then based on our behavior or what we want to focus on, it interprets this map and then focuses our oculomotor outputs to that goal-directed behavior. HBM, like all 8 parcellations, important for visuospatial working memory. 9M monitors lots of different pieces of spatial information. And like the 10s on the frontal pole, where they're active during complex tasks, 10R is also active during stimulus-orientated attention. 10V is thought to integrate all the information from the medial frontal lobe and the orbitofrontal lobe to actually help us make a behaviorally based decision. Okay, the final part of the frontal lobe that we'll talk about is the orbitofrontal region, and that has 5 parcellations. Some of these areas are interesting because they actually derive from the paleocortex that we talked about last week from the architectonics. But in essence, we've got corpus callosum connections, an IFOF connection to the occipital lobe. And we've also got, again, that uncinate fasciculus connecting the frontal pole to the temporal pole. If we look at these parcellations as a whole, they're very important in guiding our decisions, emotion, and behaviors based on olfactory information, gustatory information, and visceral. So if we look in a little bit more detail, if we look at this OFC area sitting near that gyrus rectus, it assigns reward and punishment in relation to a lot of our stimuli that we're coming across. A POFC, what that does is it evaluates the importance of taste and odor in our environment. And based on the importance of that, it guides our behavioral decisions. So if we're really hungry and there's a huge buffet in front of us, it's the parcellation which decides if we're going to keep control or sort of pig out on the buffet. 11L and 13L, integrating a lot of the olfactory and gustatory information and looking at our internal state, if we're full or not, if we want to eat more or not eat more. And then that helps us make a decision if we're going to go towards our food source or not. So I like to think of them as the gluttony parcellations. Finally, 47M, again, it's responsible like in decision making and regulating our emotions based on integrating information from all aspects of the frontal lobe. All right, so I've talked a lot, and I'm sure most of our heads are spinning. There's a lot of parcellations. It's not very manageable information. As Mike Shagru told us in the first talk, when he talked to Matt Glasser doesn't know these parcellations. But what I'm going to do is I'm just going to try and just tease out a few meaningful facts in a take-home message when we're looking at the prefrontal lobe. So if we think about it as an upside-down pyramid, we have a medial face, a dorsolateral face, and an orbitofrontal face. And based on that, we can break down the different elements of function. So on the whole, the dorsolateral face is responsible for working memory and executive function. And if we think of our central executive network, a lot of the parcellations are situated here. If we look at the medial face, that's responsible for motivation. Again, a lot of the default mode network parcellations are sitting on the medial side. And if we look at the orbitofrontal face, that's responsible for emotional regulation and judgment. So we've got 3 different faces that are roughly doing 3 different things. And if we want to summarize the tracts, the medial face is guided by the cingulate bundle, which makes sense because that's where the cingulate lobe lies. The dorsolateral face is connected by the superior, the SFL. And then the IFOF, the inferior frontooccipital fasciculus. Finally, the tracts of the orbitofrontal gyrus are the IFOF again, that one, and then the uncinate fasciculus. What's very interesting is our frontal lobes are widely connected to other parts of the brain because they're wanting to receive sensory information and also output information in how we behave and how we output through our motor systems. But for 3 surfaces of the frontal lobe that are sort of undertaking very important tasks, there's very little connection between the 3 surfaces of the brain. Sure, there's U fibers that connect the different parts of the brain, but there's no great fasciculi that join the 3 parts. And if we think of the brain in terms of networks, and we discussed small world networks last week, this is not very efficient. So it's a very interesting question how this has evolved that way. If we look at some comparative anatomy, what's clear, and all of us know, when we look at vertebrate evolution, the prefrontal cortex of primates has demonstrated explosive growth. It's what differentiates us and gives us ability. Now, what becomes interesting is if we start looking at humans and non-humans. When you look at humans versus non-human primates, the actual gray matter is largely unchanged. So the 6 layers is fairly established, and the number of cells is largely unchanged. But what is drastically different is the amount of white matter. The white matter has shown a huge volumetric expansion in the prefrontal cortex of humans versus non-humans. And interesting in this study, what they did is they compared human connections to monkey connections. And what becomes very obvious when we're looking at the white matter volume for the different types of white matter tracks, whether it's association, commissural, or projection, we as humans have much higher volumes of white matter compared to the equivalent non-human primates. What becomes also very interesting is when you're looking at where does all this white matter go from the frontal lobe in terms of to other parts of the brain. When you look at the monkey brain, only about 40% of the white matter tracts of the monkey originate from the frontal lobe to other areas, whereas in the human, it's close to 60%. So there's been an explosive shift in the amount of connections from the frontal lobes to the rest of the brain. And then when we're looking at the different tracts, it becomes very obvious that some of the tracts have evolved a lot more than others. So if we look at the cingulum and the uncinate, so that limbic system, so the factors that drive emotion and behavior, when we compare ourselves to monkeys, those tracts haven't changed much. But when we're looking at all our intelligence tracts, so the SLF, the arcuate fasciculi, and we compare those to monkeys, they've actually demonstrated explosive volumetric growth. So those tracks are very important. What they've done is they've made a much more complicated network and given us a lot more parallel processing power. Finally, just looking at some of the tracts out of interest. So the corpus callosum, when you compare our corpus callosum to non-human primates' corpus callosi, our motor system is much more posterior in that body of the corpus callosum than what it is in the non-human primates. And the reason for that is because our prefrontal areas have exploded in white matter, connecting each other on both sides. And as a result, the motor system has had to shift backwards. When we look at developmental conditions where people are born without a corpus callosum, what we actually see is commissural compensation. So what's interesting is in non-human primates, they have very developed anterior commissures. So when we're looking at our white matter volume in humans, the anterior commissure only takes up about 4%, whereas when we're looking at a monkey equivalent, it takes up 28%. So in human cases, when there's children born without a corpus callosum, the anterior commissure takes up that slack. And then they form a whole new bundle called the Probst bundle, which is thought of the superior frontooccipital fasciculus. And it's definitely been associated with conditions such as autism, schizophrenia, ADD, etc. When we look at the SLF arcuate fasciculus, that fasciculus that joins the frontal, temporal lobes and parietal lobes, monkeys have a significantly reduced volume. So this is a well-developed fasciculus that gives us our language ability. Interestingly, 60% are lateralized to the left of the arcuate fasciculi, whereas in 20% of people, they lateralize to the right, and 20% of people show bilateral representation of the arcuate fasciculi. Predominantly in females. So females tend to have bilateral arcuate fasciculi, whereas males tend to have a unilateral arcuate fasciculus. And it may explain some findings which are showing that after a left-sided stroke, females actually recover their language functions better than what males do. Potentially, it's because they've got a bilateral arcuate fasciculus and they can rewire their connectome for language function. On the right side of left dominant people, the arcuate has been shown to be important in music competence. And then just some interesting facts that uncinate fasciculus that we talked about connecting some of those areas in the frontal lobe, its damage is actually associated with the severity of antisocial traits in people that show psychopathic tendencies. So that connection between the frontal pole and temporal pole is very important behavioral properties. Finally, that inferior frontal occipital fasciculus that we talked about is almost uniquely human. So in many primates, that IFOF is undetectable. So this pink tract that connects the frontal lobe to the occipital lobe. And it's definitely has a misunderstood function at present, but it's definitely giving us some uniquely human properties. Why the frontal pole needs to directly connect with the visual cortex, primary visual areas, is unknown. Perhaps it's trying to limit the outflow of information from the visual cortex to the frontal areas, almost like a feedback mechanism to allow our brains to cope with the amount of visual information that the primary visual cortex receives. All right, so I'm going to end the talk with the motor and the premotor and the sensory cortex areas. So thankfully, I think some of these parcellations will be more known to many of you. So if we're going to break this into subregions, we're going to look at the primary motor and sensory area. We're going to look at the paracentral lobule. We're going to look at the supplementary motor area and then the premotor area. Okay, so our primary motor and sensory cortex areas have 5 parcellations, 4 motor area, then 3A, B, 1, 2, the sensory areas that I'm sure we all know about. We can see that 4 actually takes up only the posterior half of the precentral gyrus and not the entire half, because that's taken up by premotor cortex. And it also sinks deep into that central sulcus. 3A is almost a parcellation entirely within the central sulcus. So again, just shows that the organization of the brain doesn't really care in terms of what's a sulcus and a gyrus. Connectivity-wise, as we all know, it's connected to the corticospinal tract. There's areas that are connected to the superior longitudinal fasciculus, that large tract connecting all the lobes. And then we've got a very large corpus callosum connection, so bilateral connectivity of the somatosensory areas, which we saw in the previous weeks through the large somatosensory network, which functions as one conglomerate. So what does it do? So obviously 4 is primary motor area, so fine control of muscles. Interestingly, 4 has a role in visual learning during early development. 3A and 2 have relevance in terms of sensory input from deep tissues, and 3B and 1 are important for localizing tactile stimuli. 1 and 2 have much larger receptor fields than the 3A and B, and they also integrate information better bilaterally than what 3A and B do. Okay. The next part we'll talk about is the paracentral lobule, which is 4 parcellations. 4 we've already talked about. What's very interesting is that the primary motor cortex dips down into that paracentral lobule, but none of the sensory cortex does. I only discovered this after studying connectomics. So that primary sensory cortex stops shy of the midline, whereas 4 medially, that paracentral lobule is predominantly motor in function. Connectivity-wise, again, connected to the corticospinal tract, the cingulate bundle, and the corpus callosum connecting the areas bilaterally. And we've also got the 3-5 areas, 5MV, 5M, and 5L. So what do they do? So 4 we talked about, fine motor control. So 5L, what that does, it's responsible for goal-oriented hand movements based on visual stimuli or visual clues. Whereas 5M, again, it controls our hands, but it's involved when we're using sensation. So, you know, if a computer's under your bed and you're trying to feel for it and you're using your hand to feel for it, it's 5M which actually fires. And to no surprise that these 2 parcellations, they're dedicated to bimanual dexterity. And it's important they're in the midline because they're going to connect to the 5M and 5L on the other on the contralateral hemisphere. So these two parcellations are involved in bimanual dexterity. 5MV, it's shown to actually increase in activity when we're imitating a body action or a new learned procedure that we're trying to imitate, and we're actually succeeding. So the more successful our imitation is, the more active this parcellation becomes. So the supplementary motor area is the next region we're going to talk about. So if we look at the connections, we're talking about the corpus callosum connecting both SMA areas, corticospinal tract, and then also the FAT, which connects the inferior to Broca's area and the inferior frontal gyrus. So the SMA, as we all know, it's a critical relay center for the initiation and the actual production of movements, i.e. speech. And that's why people talk about the SMA syndrome in terms of when people become mute and they develop contralateral hemiplegia. And you can actually understand this from the tracts. So if you have contralateral hemiplegia, you're using the corticospinal tract. And if you're mute, is because you're not using that FAT to connect to Broca's areas and initiate speech. But interestingly, they're involved in other aspects. So SFL is part of the language network. 6MP is part of the somatosensory network, as we expect. And SFL has shown some activity during social interaction. Finally, we're going to look at the premotor area. So this has 7 parcellations. Superiorly, so this is the superior frontal gyrus, that's the inferior frontal gyrus. We've got our central sulcus and our precentral sulcus, and it's in this precentral sulcus where this premotor area sits. So we've got 6A and 6D. And then inferiorly, we've got 6 rostral and 6 ventral, so 6R and 6V. And they actually split by 3 parcellations, FEF for frontal eye field, 55B, and prefrontal eye field. So it's almost like the premotor area is split by 3 parcellations, which I will discuss. It's got lots of different connections. So these parcellations are connected by the corpus callosum body. They're connected by the SLF, which we've talked about, FAT, and the corticospinal tract. Function-wise, being a premotor area, it's important in preparing the movements that we want to do, so making them into a coherent plan. But interestingly, there's other these parcellations are involved in other activities. So 6A is active during maths, social interaction, and also when we do comparison tasks. 6R is a classic premotor area that we've talked about, and 6V is also related to Broca's area. Now, FEF, 55B and PEF, which splits the premotor area, are the most variable parcellations in the human connectome. So when you look at variability between parcellations in the entire connectome, these are the most variable. So sometimes 55V is split into 2, and there's other sorts of variations as well. What FEF does is it creates a salient map, which allows us to direct our visual attention. And it's active when we change our attention from 2 visually fixed points and also in smooth pursuit. 55B is responsible for our laryngeal muscles. So it's motor cortex for laryngeal muscles, which are also involved in speech. What PEF does, it's involved for rapid saccades, so rapid eye movements. So in the old nomenclature, these areas are thought as the frontal eye fields. Now, why evolutionary? We've got a language area between 2 frontal eye fields. Is it responsible for reading, directing gaze towards reading? No one knows, but it's actually connectomics that allowed us to discuss this, allowed us to discover this 55B parcellation. Now, if we look at models in terms of how the premotor area works, one popular model is a 2-stream motor planning hypothesis, which breaks down the premotor area into a dorsal premotor area and our ventral premotor area. So our dorsal premotor area is 6A and D, and our ventral premotor area is 6R and V. Now, the connections become interesting, and so does the clinical presentations. So if we've got a lesion in the superior part of this system, so our dorsal premotor area, the problems that we experience is visual guidance of hand movements. And how that manifests is optic ataxia. So you've got the visual information, but it's not possible for you to make a plan with your hands based on the visual information to complete a task. And the pathway also makes sense because from this dorsolateral prefrontal cortex, we've got SLF1 and 2 connections to the parietal lobe. And we'll see later on that the parietal lobe is a binding cortex. What it does is it brings auditory information, visual information, proprioceptive information to create a unique sensory experience. So that's why it's thought that people with this problem get optic ataxia. Whereas lesions in the ventral system create problems with manipulating tools or grasping tools and not being able to use them. And the connections of this are based on the arcuate fasciculus predominantly. And that's got potentially, that you can potentially interpret that we're not receiving semantic information to tell us how to use a tool or what does it mean to use a tool. So this is it was a whirlwind tour of the parcellations, and it's, again, I think what these later webinars going through the parcellations are meant to do is just show us how much connectomic data has advanced in allowing us to look at the brain in different ways. And if we can break the brain into so much detail, and we've got tools to compare it to normal brains, we can get much richer data on our patients and actually give justice to the problems that they have. Because, you know, we're never going to understand why someone has a mood disorder by saying it's a DLPFC problem. You know, it's a much more complex problem than that. And as Mike Shigru always says, if the brain was simple to understand, we would be too simple to understand. So the next webinar is going to look at the insula, the opercular, and the temporal lobe, and some of the findings through connectomics. Thank you.
Awesome. Thank you so much, Chris. It was absolutely incredible. This is my first session actually properly attended. An absolutely brilliant session. Thank you. I think, yeah, so we have finished a bit ahead of time. Actually, we just, and you have shared the session as well. Just as a reminder, if I can just ask everybody just to please register via the link directly yourselves. It just helps us with the CPD points at the end. And I will also upload the recordings all onto YouTube, as I've already done for the last 2 sessions as well. But otherwise, I guess, thank you again, everybody, and have a good evening. Thanks, Chris.
Thank you.